Welcome to Softly Curious. Tonight, we begin with something small enough to rest on a fingertip, yet precise enough to rival engineered machinery. A honeybee does not appear dramatic at first glance. It does not roar or tower. It works, and its body is built entirely for that purpose. A honeybee's surface is not smooth. It is covered in fine, branched hairs that turn the entire body into a living brush. These hairs are not decoration. As the bee moves through air, friction creates a slight electrostatic charge. Pollen grains, which are light and dry, are drawn toward that charge and cling to the hairs almost automatically. The bee does not need to aim for pollen. The pollen finds the bee. Along the hind legs, this loose dust is gathered and pressed into a more deliberate structure known as the corbicula, often called the pollen basket. It is a shallow, smooth cavity bordered by stiff hairs. With rhythmic leg movements, the bee compacts pollen into small, dense pellets. When filled, these pellets appear as bright yellow or orange saddlebags attached to the legs. The design is economical. Nothing spills. Nothing wastes motion. Above the compound eyes sit three small, simple eyes known as ocelli. These do not form images. They measure light intensity. Their function is stabilizing. As the bee flies, the ocelli help detect changes in brightness, allowing rapid adjustments in orientation. Even slight shifts in sunlight are registered. The sky becomes a reference surface. The antennae extend forward like articulated sensors. They are segmented, flexible, and densely packed with chemoreceptors. A chemoreceptor is a sensory structure that binds to specific chemical molecules. Through these receptors, the bee detects pheromones, subtle chemical signals that indicate identity, condition, or state. It can distinguish floral scents at remarkable sensitivity. Odor is not background information. It is structure. At the front of the head lies the proboscis, a long, extendable feeding organ. It is often described as a tongue, but it functions more like a straw combined with a sponge. When extended into a flower, it draws nectar upward through capillary action and muscular pumping. The mechanism is precise and efficient. The bee does not chew nectar. It collects it in liquid form. Flanking the proboscis are the mandibles. These are strong jaw-like structures used not for eating nectar, but for shaping and manipulating materials. Mandibles cut, mold, and carry. They can knead wax, grip surfaces, or adjust structural elements. In a body only a few centimeters long, mechanical tools are embedded seamlessly into anatomy. The wings, nearly transparent, appear fragile. They are not. A honeybee beats its wings approximately 200 times per second. This rapid oscillation produces lift through a figure-8 motion that maximizes aerodynamic efficiency. The sound we recognize as a hum is the byproduct of that rhythm. Beneath the wings lie powerful indirect flight muscles. Rather than attaching directly to the wings, these muscles deform the thorax itself. As the thoracic box flexes, the wings are driven up and down. This arrangement allows extraordinary energy efficiency relative to body size. Fueling this motion requires metabolic precision. The bee's body operates with an open circulatory system. Instead of blood contained within closed vessels, it circulates hemolymph, a fluid that bathes internal organs directly. Hemolymph transports nutrients and hormones, but does not carry oxygen in the same way human blood does. Oxygen enters through spiracles, small openings along the body, and travels through a network of tubes called trachea. Gas exchange occurs without lungs. Air flows inward, diffuses, and exits in quiet cycles. Development into this form is not gradual growth, but transformation. A honeybee begins as an egg, laid upright in a wax cell. Within days, it becomes a larva, a soft, legless form devoted entirely to feeding. The larva molts repeatedly as it increases in size. It then enters the pupal stage, enclosed and reorganizing. During pupation, tissues dissolve and reform. Wings develop, eyes darken, legs articulate. This process is called complete metamorphosis, meaning a full structural reorganization between life stages. The adult that emerges is not an enlarged larva. It is a rebuilt organism. Among adult bees, physical differences mark distinct biological roles. The queen is longer, with a more elongated abdomen designed for egg production. Her ovaries are fully developed. Worker bees are smaller, with specialized structures such as pollen baskets and wax glands. Male bees, known as drones, possess larger eyes that nearly meet at the top of the head, increasing visual sensitivity during flight. They lack stingers. Their bodies are tuned for a singular reproductive function. The stinger itself is a modified ovipositor, an egg-laying organ that has evolved into a defensive instrument in worker bees. It is barbed. When embedded in thick skin, those barbs anchor it in place. The act of stinging can be fatal to the worker because the stinger detaches along with part of the abdominal tissue. The mechanism injects venom through a muscular pumping action even after separation. It is an anatomical trade-off between defense and survival. Inside the head, the brain weighs less than a milligram, yet it contains roughly 1 million neurons. This number is small compared to a human brain, but density and organization matter more than size alone. Neural clusters called mushroom bodies are involved in learning and memory. Sensory information is integrated rapidly. Decision-making circuits operate within tight energetic constraints. In 2006, scientists completed the sequencing of the honeybee genome. A genome is the complete set of genetic instructions within an organism. The analysis revealed an unexpectedly refined set of genes related to social communication and immune response. Compared to some insects, honeybees possess fewer detoxification genes, which may partly explain their sensitivity to certain environmental chemicals. At the same time, genes linked to olfactory reception are highly developed, reflecting the central role of scent in perception. When viewed as a whole, the honeybee's body is not ornamental. Every surface, every joint, every filament has purpose. It is compact without excess. It senses wavelengths we cannot see, scents we cannot detect, and vibrations too subtle for our perception. It transforms liquid into stored energy, dust into structure, and air into motion. There is no wasted architecture here, only refinement. Inside a honey bee, regulation is constant. Nothing is left to chance. Temperature, chemistry, energy, timing, all are quietly adjusted in rhythms too small to notice from the outside. A honeybee does not rely only on the warmth of the sun. It can generate heat from within. By rapidly contracting its thoracic flight muscles without moving its wings, it produces warmth through vibration. This is thermogenesis, meaning heat production through muscular activity. Even at rest, a bee can raise its body temperature above the surrounding air. Flight muscles function not only as engines, but as internal furnaces. Within the hive, stability matters even more. Brood development requires a narrow thermal window, typically between 34 and 36 degrees Celsius. Deviations of just a few degrees can slow development or affect survival. Workers respond collectively. Some cluster tightly around brood cells, forming a living insulating layer. Others vibrate their muscles to add warmth. Temperature inside the hive is not accidental. It is maintained with precision comparable to engineered incubation systems. When heat rises too high, the strategy reverses. Workers line the entrance and fan their wings in coordinated pulses. The movement is not random. It directs airflow through the comb structure, drawing warm air out and pulling cooler air in. This ventilation behavior reduces internal temperature and regulates humidity at the same time. A hive breathes. Water becomes part of this regulation system. Foragers collect droplets and deposit them inside the hive. Other workers spread the water across cell surfaces. As it evaporates, it absorbs heat, producing a cooling effect similar to perspiration. Evaporative cooling is a physical process in which liquid absorbs energy as it changes into vapor. Even in high summer, internal balance is maintained through this subtle exchange. Nutrition is equally specialized. Young worker bees possess hypopharyngeal glands, structures located in the head that produce royal jelly. Royal jelly is a nutrient-dense secretion rich in proteins, lipids, and vitamins. It is fed to larvae and, in larger and sustained quantities, to developing queens. The production of this substance is age-dependent. As workers mature, gland activity changes, and their physiological roles shift accordingly. Lower on the abdomen, wax glands operate with equal precision. These glands are located on the underside of specific abdominal segments. They secrete liquid wax that solidifies into small translucent scales, often described as wax plates. A worker uses her hind legs to remove these scales and passes them forward to the mandibles, where the wax is softened and molded. The raw material of architecture emerges from metabolism itself. The conversion of nectar into honey is another internal transformation governed by enzymes. Nectar is primarily composed of sucrose, a disaccharide made of glucose and fructose. Inside the honey's stomach, an organ separate from the digestive stomach, nectar mixes with the enzyme invertase. Invertase breaks sucrose into its simpler components, glucose and fructose. This enzymatic hydrolysis, meaning chemical breakdown using water, alters both sweetness and stability. Back in the hive, further dehydration reduces water content to below approximately 18%, limiting microbial growth. What began as a dilute plant secretion becomes a concentrated, long-lasting energy reserve. The digestive system itself is partitioned efficiently. The honey stomach stores collected nectar without digesting it. The midgut performs nutrient absorption for the bee's own metabolism. This separation allows a forager to carry large volumes of nectar without consuming it. Transport and nutrition are distinct processes, managed in parallel. Immune defense operates without adaptive antibodies like those found in vertebrates. Instead, honeybees rely on innate immunity, meaning a built-in, generalized defense system. Antimicrobial peptides are produced in response to infection. These short proteins disrupt bacterial membranes. Hemocytes, cells circulating in hemolymph, can engulf pathogens in a process called phagocytosis. Although their immune gene repertoire is smaller than that of some other insects, the system is efficient within the ecological niche they occupy. Perception and physiology intersect in subtle preferences. Many flowers display ultraviolet patterns invisible to humans, but highly distinct to bees. These patterns, sometimes called nectar guides, direct landing and feeding behavior. Experimental studies show that bees learn to associate ultraviolet contrast with higher nectar reward. Preference is shaped by both sensory detection and energetic return. Efficiency becomes memory. Flight range is determined by energy budget. A typical honeybee may forage within a radius of 2 to 3 kilometers, though longer flights are possible under certain conditions. Each flight consumes measurable metabolic energy. Sugars from honey are oxidized to produce ATP, adenosine triphosphate, the molecule that stores and transfers cellular energy. Oxygen consumption increases sharply during sustained flight. Metabolic rate during flight can be many times higher than at rest. Energy is spent carefully. Foraging decisions reflect that constraint. Respiration occurs through spiracles along the body wall. Air enters and travels through branching tracheal tubes, delivering oxygen directly to tissues. There is no lung to inflate. Instead, abdominal movements assist ventilation. The rate of oxygen consumption correlates with muscular demand. During thermogenesis or flight, respiratory exchange accelerates. Inside the brain, plasticity allows adaptation. Neural plasticity refers to the capacity of neural circuits to reorganize in response to experience. In honeybees, exposure to complex environments and repeated foraging routes can alter synaptic strength within the mushroom bodies. Learning modifies structure at microscopic scales. Even with roughly 1 million neurons, flexibility exists. Time is also regulated internally. Honeybees possess circadian rhythms, biological cycles of approximately 24 hours. These rhythms are controlled by internal molecular clocks involving feedback loops of gene expression. A circadian rhythm is an endogenous, meaning internally generated, cycle that persists even without external cues. Bees can anticipate the opening times of certain flowers. They arrive not randomly, but predictively. Internal timing aligns physiology with environment. Taken together, these processes form a network of adjustments. Heat is generated or released. Nectar is altered chemically. Wax is secreted and shaped. Pathogens are neutralized. Oxygen flows without lungs. Memory shifts with experience. Timing unfolds with quiet regularity. Nothing inside the honeybee is idle. Each internal system supports continuity, precision, and balance. The body does not simply exist, it regulates. A single honeybee can be understood by studying its body. A colony, however, must be understood as a system. It is not a crowd, it is an arrangement. At the center of this arrangement stands the queen. She does not command in the human sense. She does not issue instructions. Her influence is chemical. The queen produces pheromones, specialized signaling molecules that circulate through the hive as a kind of invisible architecture. A pheromone is a chemical message released by one individual that affects the behavior or physiology of others of the same species. In a healthy colony, the queen's mandibular pheromone maintains social cohesion. It signals presence, fertility, and stability. Workers that detect this signal remain behaviorally aligned. The colony does not follow her voice. It follows her chemistry. Around her, thousands of workers move through a predictable sequence of roles. Age determines duty in a pattern known as temporal polytheism, meaning task allocation based on age. A newly emerged worker begins her life inside the hive. During her first days, she cleans cells, preparing them for new eggs. Cleanliness is not cosmetic. It reduces microbial growth and supports larval survival. Hygiene is foundational. As the worker matures, her internal physiology shifts. Glands activate. She becomes a nurse, feeding larvae with secretions rich in nutrients. Larval care is precise. Each larva is visited repeatedly and fed according to developmental stage. The brood is not left unattended. Its growth is monitored continuously. Later still, the same worker may shift to comb building or food processing. She receives nectar from returning foragers and passes it along, contributing to its transformation. Eventually, in the final phase of her life, she becomes a forager. This outward transition is irreversible. Once she begins flying regularly outside the hive, she does not return to nursing. The colony allocates labor through biology rather than instruction. Male bees, known as drones, occupy a different biological niche. They do not collect nectar. They do not produce wax. They lack stingers and do not participate in defense. Their primary function is reproductive. Drones are produced from unfertilized eggs through a process called haplodiploidy, meaning males carry only one set of chromosomes. Their large eyes enhance visual tracking during flight, a feature linked to their role in mating. Reproduction occurs during mating flights, when a virgin queen leaves the hive and flies to congregation areas where drones gather. In midair, mating takes place. The drone's reproductive organ everts and transfers sperm before detaching. The drone dies shortly afterward. It is a singular biological event. The queen does not mate once. She mates multiple times, often with a dozen or more drones during her mating period. This polyandry, meaning multiple mating partners, increases genetic diversity within the colony. The sperm she collects is stored in a specialized internal organ called the spermatheca. There it can remain viable for years. From this reservoir, she selectively fertilizes eggs throughout her lifetime. Genetic diversity within the worker population affects colony resilience. Colonies with higher genetic variation tend to show improved disease resistance and more flexible behavioral responses. Different patrilinees, meaning groups of workers sharing the same father, may vary slightly in sensitivity to stimuli or thresholds for certain tasks. Variation becomes strength. The physical structure that houses this social system is equally deliberate. Honeycomb is built from beeswax into a repeating pattern of hexagonal cells. The hexagon is not aesthetic preference. It is a geometric solution. A hexagonal tiling covers a surface without gaps while minimizing perimeter length for a given area. This means less wax is required to enclose maximum storage space. The geometry balances strength and material efficiency. Wax walls meet at precise angles, creating a lattice capable of supporting honey, brood, and the weight of the colony itself. Within this structure, decisions emerge collectively. No single bee surveys the entire hive. Yet when a colony must relocate, it does not fragment into chaos. Scout bees explore potential nesting sites. Each scout evaluates factors such as cavity volume, entrance size, and internal dryness. Upon returning, she communicates her assessment through dance behavior. The intensity and duration of the dance correlate with perceived site quality. Other scouts visit the advertised location. If they agree, they reinforce the signal with their own dances. If not, the signal fades. Through repetition and recruitment, one site eventually gains majority support. This process resembles voting. There is no ballot, but there is accumulation of preference. Consensus forms gradually. Taken together, the colony functions as a distributed system. Authority is not centralized in a single nervous system. It is diffused across thousands of individuals responding to local information. Chemical gradients, age-linked physiology, and structural design integrate into a coherent whole. The queen stabilizes through pheromones. Workers rotate through life stages according to internal timing. Drones fulfill a brief but essential role. Wax becomes geometry. Scouts become voters. Guards become filters. The hive is often described as a superorganism, meaning a group that functions as a single biological entity. This term reflects more than metaphor. Metabolism, reproduction, defense, and decision-making operate at the colony level. Individual bees are components, each with limited scope. The system emerges from their interaction. Inside the hive, there is no visible hierarchy in motion. There is rhythm instead. Roles shift with age. Signals diffuse through air and contact. Geometry supports biology. Reproduction distributes variation. Decisions arise without debate. The logic of the colony is quiet but exact. It does not depend on command. It depends on alignment. A honeybee colony does not remain static. Growth carries consequence. When population density increases and internal space tightens, a threshold is crossed. The response is not random dispersal. It is swarming. Swarming is the process by which a colony divides into two living systems. It is not collapse. It is reproduction at the colony level. As numbers rise and resources accumulate, a portion of workers prepare to leave with the existing queen. They gorge on honey before departure, filling their honey stomachs to capacity. This stored energy fuels the transition. Then, in a sudden coordinated movement, thousands of bees pour from the hive and gather on a nearby branch or surface, forming a dense, temporary cluster. This suspended mass is not disordered. It is a transitional state. While clustered, scout bees begin searching for a permanent nesting site. The swarm may hang for hours or days, adjusting position as scouts return with information. During this period, the colony is exposed, yet it remains composed. Inside the original hive, a different transformation is underway. New queens are being produced. Special cells are constructed vertically, elongated, and peanut-shaped, distinct from the horizontal worker cells. These are queen cells. Their architecture allows for the development of a larger abdomen and full reproductive organs. Shape here is destiny. Emergency queen production follows a different trigger. If a colony loses its queen unexpectedly, workers respond by selecting several very young larvae and modifying their developmental pathway. By feeding them sustained quantities of royal jelly, they alter gene expression and physiology. The same genetic blueprint can yield a worker or a queen, depending on nutritional input. Development is plastic, meaning flexible within biological limits. The balance of pheromonal signals within a hive stabilizes organization. When that balance is disrupted, behavior shifts rapidly. A sudden reduction in queen pheromone can lead to agitation, accelerated cell modification, and heightened responsiveness. Chemical absence creates behavioral acceleration. The system reacts to gradients rather than commands. Colonies also exhibit measurable stress responses. External pressures such as temperature extremes, resource scarcity, or chemical exposure can alter activity patterns. Foraging intensity may increase or decrease. Brood production may slow. Worker lifespan can shorten under sustained strain. These changes are not emotional in the human sense. They are physiological adjustments reflected in collective output. One striking aspect of colony health is the removal of compromised individuals. Bees infected with certain pathogens or parasites may display altered chemical cues. Nestmates detect these shifts through antennal contact. In some cases, affected individuals are forcibly removed from the hive. This behavior reduces pathogen load. It is a form of social immunity, meaning group-level disease defense arising from coordinated action. Hygienic behavior extends further. Workers inspect brood cells and can detect subtle abnormalities beneath the wax cap. If a developing pupa is infected, workers uncap the cell and remove the contents. This interrupts pathogen replication cycles. Selective breeding programs have identified colonies with particularly strong hygienic traits, demonstrating that these responses vary genetically. Coordination within the hive is not limited to chemical cues. Vibrational communication also plays a role. Bees can transmit signals through comb substrate by producing short bursts of thoracic vibration. These signals influence activity levels of nearby workers. A brief vibrational pulse may prime individuals to increase task engagement. Communication travels not only through air, but through structure. Over time, patterns of resource locations and seasonal shifts accumulate as distributed knowledge. Although no single bee stores a complete map, the colony maintains what can be described as a form of collective memory. Collective memory refers to information retained at the group level through overlapping individual experiences. As older foragers die and younger ones take their place, learned routes in profitable locations persist through repeated recruitment and reinforcement. Season introduces another transformation. As temperatures fall and floral resources decline, activity contracts inward. The colony forms a winter cluster. Workers gather tightly around the queen and remaining brood, creating a living sphere. Bees on the outer layer act as insulation, while those inside generate heat through muscular activity. Individuals rotate positions gradually, preventing prolonged exposure to cold at the surface. The cluster shifts slowly across stored honey reserves as winter progresses. During this period, consumption is calculated. Although bees do not perform arithmetic, their behavior reflects estimation of available stores relative to population size and season length. Reduced brood production lowers energy demand. Foraging ceases in cold climates. The colony survives on accumulated honey, metabolized carefully. If stores are insufficient, starvation can occur even within a structurally intact hive. These seasonal and structural adjustments reveal regulation at scale. Swarming redistributes abundance. Emergency queen development restores reproductive continuity. Hygienic removal reduces pathogen spread. Vibrational signals subtly increase or decrease task intensity. Winter clustering conserves metabolic heat while rationing stored honey. None of these responses rely on central command. They emerge from thresholds and feedback loops. When population density rises, division begins. When reproductive signals weaken, new queens are prepared. When chemical traces of disease appear, removal follows. When temperatures fall, bodies gather into warmth. Stability is not fixed, but continuously recalibrated. What appears sudden from the outside is usually the result of accumulation. A swarm's departure reflects weeks of growth and preparation. A winter cluster reflects months of resource calculation. Intelligence within the hive is distributed rather than localized, expressed through interaction, repetition, and adjustment. Continuity depends on change, and balance is maintained through motion. The colony does not stand still to survive. It adapts quietly and precisely. A honeybee does not speak with sound in the way mammals do. Inside the hive, communication moves through motion, scent, angle, and vibration. Information is carried in patterns rather than words. The air itself becomes a medium for direction. One of the most studied forms of animal communication is the waggle dance. When a forager discovers a profitable nectar source, she returns to the hive and performs a repeated figure-8 movement on the vertical comb surface. During the central waggle run, she vibrates her abdomen while moving forward in a straight line. The angle of this line relative to vertical corresponds to the angle between the sun and the food source outside. Vertical upward means fly toward the sun. A deviation to the right or left encodes a deviation from the sun's position in the sky. The duration of the waggle run correlates with distance. Longer runs signal farther destinations. The dance occurs in darkness. Inside the hive, vision is limited. Yet workers perceive the dancer's movements through touch and vibration. They follow closely, contacting her body with their antennae, decoding angle and duration through repeated exposure. Information is transferred through repetition and proximity. To calculate direction, the honeybee relies on the sun as a compass. Even as the sun moves across the sky during the day, bees compensate for its shifting position. This requires an internal time sense, allowing adjustment for solar movement. Direction is not fixed. It is recalculated continuously based on celestial reference. When clouds obscure the sun, navigation does not collapse. Honeybees detect polarized light patterns in the sky. Polarized light refers to sunlight scattered in a specific orientation by atmospheric particles. Although invisible to humans without filters, these patterns remain detectable to bees. Specialized photoreceptors in the compound eyes respond to polarization angles, allowing continued orientation even under partial cloud cover. The sky retains structure. Distance measurement is achieved through optic flow. As a bee flies, objects pass across her visual field. The rate at which visual patterns move across the retina provides information about distance traveled. This process, known as visual odometry, translates motion into metric estimation. A bee flying low over textured terrain perceives rapid optic flow and estimates a longer journey than one flying higher over uniform ground. Distance is sensed through movement itself. Once a profitable flower patch is identified, many honeybees exhibit what researchers call flower constancy. This means a forager may repeatedly visit the same plant species during a single trip, even if other flowers are available. Such constancy improves pollination efficiency and reduces cognitive switching costs. Memory and reward align. Chemical signals support this navigational system. The Nasanov pheromone, released from a gland near the tip of the abdomen, serves as an orientation cue. When bees expose this gland and fan their wings, they disperse the pheromone into the air. It signals location, guiding nestmates toward the hive entrance or a newly occupied site. Scent extends direction beyond sight. Foragers also leave subtle scent traces on flowers they visit. These marks can deter immediate revisits by others, preventing redundant foraging. Odor becomes a temporary annotation on the landscape, reducing overlap and increasing efficiency. At the hive entrance, returning bees align themselves using familiar visual markers. Landmarks such as tree trunks, fence lines, or contrasting shapes help refine approach trajectories. Experiments have shown that bees displaced to unfamiliar locations can still orient homeward, using learned landmarks combined with sun compass information. Navigation integrates multiple cues. Flight range varies with resource availability, but honeybees can travel up to 5 kilometers from the hive when necessary. Such journeys demand substantial energy and accurate memory. Long-distance flights expand the effective territory of the colony, mapping floral distribution across broad areas. Mapping is not stored as a single internal chart. Instead, repeated flights reinforce routes. Experienced foragers demonstrate higher efficiency, reducing search time and adjusting paths according to environmental change. The landscape becomes familiar through repetition. Not all bee species rely on the same communication architecture. Bumblebees, for example, do not perform the elaborate waggle dance of honeybees. Their colonies are smaller, and recruitment tends to be less formalized. Information sharing may occur through scent cues or simple activation of foraging behavior rather than geometric encoding. Solitary bees, which do not live in large colonies, navigate entirely through individual memory. Each female provisions her own nest and does not need to broadcast food location to others. The diversity of bee communication reflects differences in social structure. Research has also explored the possibility that honeybees respond to electromagnetic fields. Some experiments suggest that bees may detect weak electric charges associated with flowers or environmental fluctuations. Floral surfaces can carry slight electrical potentials that change after a bee visit. Whether bees use these cues systematically remains under investigation, but evidence indicates sensitivity, at least at short range. Perception may extend beyond light and scent. Within this communication network, redundancy is common. A single forager does not represent absolute truth. Multiple scouts verify sites. Multiple cues overlap. Direction from the sun aligns with optic flow. Polarized light supports orientation when direct sunlight is absent. Landmarks refine final approach. Chemical signals mark convergence points. Memory capacity supports this complexity. Honeybees can remember spatial relationships between hive and food sources across days. They adjust departure times to coincide with peak nectar production of specific flowers. Timing, direction, and reward integrate into learned routines. The communication system operates without centralized broadcast. Each bee encodes and decodes based on local information. The hive becomes a dynamic field of signals. Angles traced on wax, pheromones drifting through air, landmarks embedded in memory, polarized light etched into perception. In motion, language forms. It is measured in degrees relative to the sun, in milliseconds of abdominal vibration, in gradients of scent, in the shifting geometry of the sky. Through these mechanisms, thousands of individuals coordinate across kilometers without maps, without spoken instruction, without visible leaders. The air outside the hive is vast, yet a honeybee moves through it with calibrated precision, guided by patterns most observers never see. Navigation explains how a honeybee moves through space. learning explains how it improves. A honeybee does not respond to the world as a fixed machine. It adjusts. Experience leaves traces in neural circuits, altering future decisions in subtle but measurable ways. The brain is small, yet it is not rigid. Color perception provides an early example of this flexibility. Honeybees can distinguish between a wide range of colors, including ultraviolet wavelengths invisible to humans. Their visual system is trichromatic, meaning it relies on 3 types of photoreceptors sensitive to ultraviolet, blue, and green light. Red appears dark to them, but contrasts in blue and ultraviolet are vivid. Experiments using colored panels show that bees quickly learn to associate specific colors with sugar rewards. When reward is removed, preference declines. Learning modifies attraction. Beyond color, bees can discriminate shapes. In laboratory settings, they have been trained to differentiate between circles and triangles, horizontal and vertical lines, symmetrical and asymmetrical patterns. Shape recognition is not perfect, but it is consistent above chance. This indicates pattern processing rather than simple brightness detection. Visual features are encoded, compared, and retrieved. Studies have also explored numerical cognition. In controlled experiments, bees were presented with visual arrays containing different numbers of shapes. With training, they learned to choose panels displaying a specific quantity, such as 2 or 3 elements, regardless of the shapes themselves. Some studies suggest bees can perform simple arithmetic tasks, such as adding or subtracting 1 from a presented number of items when guided by symbolic color cues. While this does not imply abstract mathematics, it indicates capacity for low-level quantity processing. Problem-solving abilities extend into physical tasks. In one experiment, bees were trained to pull a small string to access a sugar reward located beneath a transparent barrier. Initially, they failed. Over repeated attempts, individuals learned to grasp and pull the string with coordinated movements. This sequence required understanding that pulling the string moved the reward within reach. It was not a natural foraging behavior. It emerged through trial and adjustment. Trial and error learning is fundamental to bee cognition. When encountering a novel flower type, a forager may initially make inefficient attempts to access nectar. Through repetition, motor patterns refine. Handling time decreases. Energy return improves. Mistakes are not wasted events. They are information. Learning does not occur in isolation. Social learning has been observed under experimental conditions. In tasks involving artificial puzzles, naive bees that observed trained individuals performing a successful action learned more quickly than those without exposure. Social learning refers to acquiring information by watching others. Although this capacity is limited compared to vertebrates, it demonstrates that observation can influence behavior. Olfactory memory, the memory of scent, is particularly robust. Bees can retain associations between specific odors and rewards for several days. Conditioning experiments, such as the proboscis extension reflex test, show that when a scent is paired repeatedly with sugar, bees extend their feeding organ in response to the scent alone. This learned reflex persists, indicating long-term memory formation. Odor cues in the environment thus gain predictive meaning. Neural plasticity underlies these capabilities. Plasticity means the ability of neural connections to strengthen or weaken over time. In honeybees, repeated learning tasks are associated with changes in synaptic density within the mushroom bodies, brain regions involved in sensory integration and memory. Although the total neuron count remains constant, connectivity shifts. Experience alters architecture at microscopic scale. Research on pesticide exposure highlights this vulnerability. Certain compounds, particularly neonicotinoids, act on nicotinic acetylcholine receptors in the insect nervous system. These receptors are involved in signal transmission between neurons. At high doses, such chemicals are lethal. At low doses, they may not cause immediate death, but can impair memory and orientation. Bees exposed to subalthal levels sometimes exhibit difficulty returning to the hive or reduced ability to learn new associations. The effect is subtle but measurable. Even without chemical stressors, energy limitation affects cognition. The brain consumes metabolic resources. During extended foraging or under food scarcity, glucose availability can influence neural efficiency. There is a biological cost to processing information. When a once abundant flower becomes scarce, foragers shift to new sources, updating learned associations rather than moving randomly. Short-term memory supports immediate return flights, while repeated reward strengthens long-term memory that can last for days. This transition involves changes in gene expression within neurons, allowing experience to reshape neural connections and refine future decisions. Cognitive function also changes across life stages. Younger workers performing in-hive duties show different neural activity patterns compared to experienced foragers navigating open landscapes. As bees transition to outside work, regions of the brain associated with sensory integration and memory exhibit increased synaptic complexity. Functional demand shapes neural architecture. Learning is therefore not static, but developmentally tuned. This cognitive capacity does not imply abstract reasoning, but efficient signal processing and flexible adjustment. Honeybees learn quickly when rewards are stable, adapt when conditions change, and falter under stress before recovering when balance returns. Within a brain smaller than a seed, calibration is constant. A honeybee does not intend to shape landscapes. It searches for nectar and pollen to sustain its colony. Yet in doing so, it moves genetic material across entire ecosystems. Pollination is not a side effect of flight. It is a structural force. Pollination occurs when pollen grains, produced in the male structures of a flower, are transferred to the female structures of the same or another flower of the same species. This transfer enables fertilization and seed production. When a honeybee lands on a bloom, pollen adheres to the fine hairs covering its body. As it moves to the next flower, some of that pollen brushes against the stigma, the receptive surface of the plant's female organ. Fertilization follows. Fruit and seed development begin. Without this transfer, many plants cannot reproduce successfully. Honeybees are generalist foragers. They visit a wide variety of flowering plants, making them highly effective pollinators across agricultural and wild landscapes. Their tendency toward flower constancy, repeatedly visiting the same plant species during a foraging trip, increases the probability that pollen reaches a compatible flower. Efficiency emerges from repetition. Not all pollination works the same way. Some plants, such as tomatoes and certain berries, release pollen only when vibrated at specific frequencies. This process is known as buzz pollination, meaning pollen is dislodged through rapid vibration. Bumblebees are particularly adept at this technique. They grasp the flower and activate their flight muscles without flying, producing a high-frequency vibration that shakes pollen free. Honeybees do not perform true buzz pollination in the same way. In ecosystems where such plants are abundant, bumblebees play a critical complementary role. Beyond these social species, solitary bees contribute quietly but powerfully. Unlike honeybees, solitary bees do not live in large colonies. Each female constructs and provisions her own nest. Yet many solitary species are highly efficient pollinators. In certain crops, a relatively small number of mason bees can achieve pollination rates comparable to much larger numbers of honeybees. Efficiency varies by plant structure and behavior, illustrating that pollination is not a uniform service delivered by a single species. It is a network of interactions. Modern agriculture depends heavily on this network. Crops such as almonds, blueberries, apples, and many vegetables rely on insect-mediated pollination to produce marketable yields. In large-scale almond orchards, for example, managed honeybee colonies are transported seasonally to ensure sufficient pollinator presence during bloom. This practice reflects a direct economic link between bee health and food production. Pollination is not only ecological, it is agricultural infrastructure. Wild plant communities are equally dependent. Flowering plants in meadows, forests, and grasslands rely on pollinators to maintain genetic diversity. When bees move pollen between individuals separated by distance, they reduce inbreeding and increase resilience. Wildflower populations are stabilized through repeated pollinator visits. The diversity of bees supports the diversity of plants, and the reverse is also true. However, agricultural landscapes often simplify plant diversity. Monoculture, meaning the cultivation of a single crop species over large areas, reduces floral variety. During bloom, such fields can provide abundant nectar. Outside that window, they may offer little. For pollinators, this creates nutritional bottlenecks. A diet restricted to one pollen source may lack certain amino acids or micronutrients. Nutritional diversity influences immune function and longevity. Habitat loss compounds these pressures. Urban expansion, intensive farming, and infrastructure development reduce natural nesting sites and wildflower corridors. Hedgerows, fallow fields, and unmanaged margins once provided forage continuity. When these disappear, bees must travel farther or compete more intensely for limited resources. Distance increases energy expenditure. Fragmentation reduces connectivity. Climate change introduces another variable, timing. Many flowering plants respond to temperature cues. Warmer springs can cause earlier blooming. If pollinator emergence does not shift at the same rate, temporal mismatches may occur. A temporal mismatch means that flowers bloom before or after peak pollinator activity. Even slight shifts can affect reproductive success. In some regions, long-term observations indicate altered bloom periods and changes in bee foraging patterns. Population trends are not uniform across the globe. Data collected over recent decades show regional variation. In some areas, managed honeybee colony numbers have stabilized or even increased due to active beekeeping practices. In others, losses remain significant. Wild bee populations display similarly varied patterns, with certain species declining sharply while others remain stable. These differences reflect local land use, climate, disease pressure, and agricultural practices. There is no single global trajectory. In ecological systems, redundancy supports resilience. Multiple bee species performing overlapping pollination roles reduce dependency on any single population. If one species declines in a given region, others may partially compensate. This buffering effect depends on diversity. When diversity narrows, systems become more fragile. The living world is shaped not only by visible structures, but by movement between them. Each foraging trip transfers microscopic grains that determine whether fruit will form, whether seeds will disperse, whether future blooms will appear. A bee's path across a meadow may seem small, but multiplied across thousands of individuals and millions of flights, it becomes ecological continuity. Pollination is not dramatic, it is incremental. Grain by grain, visit by visit, it sustains both cultivated fields and wild landscapes. In this exchange between insect and plant, survival is mutual. The bee seeks nourishment. The flower seeks reproduction. Together, they sustain the living world. Environmental pressure does not always arrive dramatically. It accumulates. A colony may appear stable from the outside, while subtle stressors reshape its internal balance. Among the most widely discussed phenomena in recent decades is colony collapse disorder, often abbreviated as CCD. In affected colonies, worker bees disappear abruptly, leaving behind brood, food stores, and the queen. The pattern is unusual because large numbers of dead bees are not typically found near the hive. Research suggests that CCD is not caused by a single factor, but by interacting stressors that weaken colony resilience over time. One of the most serious biological threats to honeybees is Varroa destructor, a parasitic mite that reproduces inside brood cells. The mite feeds on the bee's fat body tissue, a nutrient storage organ essential for immunity and detoxification. Beyond direct damage, Varroa acts as a vector for viruses, amplifying infection rates within colonies. Without management, heavy infestations can destabilize a hive within months. Varroa pressure is particularly severe for managed honeybee colonies, which often exist in high densities that facilitate parasite spread. Viral infections compound this challenge. Deformed wing virus, for example, can cause emerging bees to develop crumpled wings and reduced lifespan. Other viruses impair navigation, immunity, or brood development. In healthy colonies, immune responses and social behaviors may suppress viral load. Under stress, viral replication accelerates. Disease does not operate in isolation. It interacts with nutrition, genetics, and environmental exposure. Chemical exposure introduces additional complexity. Neonicotinoid pesticides, which act on nicotinic acetylcholine receptors in the insect nervous system, can disrupt neural signaling. At lethal doses, they cause paralysis. At sublethal doses, they may impair learning and orientation. A forager exposed to low levels may struggle to return to the hive. The impact is not always immediate mortality, but reduced efficiency and survival over time. Agricultural chemicals extend beyond insecticides. Fungicides and herbicides, though not designed to target bees, can interact with detoxification pathways. Some compounds weaken immune responses or alter gut microbiota, the community of beneficial microbes living in the digestive tract. A compromised microbiome can reduce nutrient absorption and disease resistance. The cumulative effect of multiple chemicals, even at low concentrations, remains an active area of research. Microplastic particles have also entered this conversation. Recent studies have detected microscopic plastic fragments in bee-collected pollen and in hive products. These particles originate from degraded packaging, synthetic fabrics, and urban dust. While the long-term physiological impact on bees is still under investigation, the presence of microplastics illustrates how deeply human material cycles now intersect with pollinator biology. Light pollution introduces another subtle disturbance. Artificial lighting at night can alter insect navigation and circadian rhythms. Although honeybees are primarily diurnal, prolonged exposure to artificial light near urban hives may disrupt rest cycles or orientation. The biological clock depends on natural light-dark cycles. When those cycles shift, timing may drift. Noise pollution has also been shown to influence pollinator behavior. Vibrational and acoustic disturbances from traffic or machinery can alter foraging patterns and stress responses. Some studies suggest that chronic noise reduces flower visitation rates or changes communication dynamics within hives. Sound, even when not consciously perceived as harmful, can modify behavior. These pressures do not affect all bee species equally. Varroa mites primarily threaten honeybees, particularly managed colonies. Habitat loss and pesticide exposure, however, impact wild bee populations as well. In some cases, certain solitary bee species are even more sensitive to chemical exposure than honeybees, lacking the buffering effect of large colonies. Urban and rural environments present contrasting risks and opportunities. Intensive agricultural regions may expose bees to higher pesticide loads and reduced floral diversity. Yet some urban areas, with parks, gardens, and varied flowering plants, can support surprisingly robust bee populations. Studies in several cities have found that certain wild bee species perform as well as, or better than, their rural counterparts when diverse forage is available. Environmental pressure is rarely singular. It is layered. Parasites weaken immunity. Viruses exploit weakness. Chemicals interfere with neural signaling. Habitat fragmentation limits nutrition. Artificial light and noise shift behavioral patterns. Yet responses vary by species, region, and management practice. The living world around bees is not static, and neither is their resilience. Within this shifting landscape, survival depends on adaptation, diversity, and the quiet capacity of systems to absorb stress without collapse. When a honeybee gathers nectar from a flower, it is collecting a solution that is mostly water. Nectar is typically composed of sucrose, dissolved in a high percentage of liquid. On its own, nectar is unstable. It ferments easily, supports microbial growth, and spoils within days. Yet inside the hive, this dilute plant secretion is transformed into one of the most chemically stable natural foods known: honey. The transformation begins before the bee even returns home. Nectar is stored in a specialized organ called the honey stomach, separate from the digestive stomach. Here, enzymes begin their work. One of the most important is invertase. Invertase is an enzyme that breaks down sucrose, a disaccharide, into two simpler sugars, glucose and fructose. This process is called enzymatic hydrolysis, meaning a chemical breakdown facilitated by water and a biological catalyst. By converting sucrose into monosaccharides, the bee alters both the sweetness profile and the chemical stability of the liquid. Back at the hive, nectar is passed from one worker to another through regurgitation and reprocessing. Each transfer further exposes the liquid to enzymes and reduces its water content. The bees spread the treated nectar into thin layers within wax cells. Surface area increases. Evaporation accelerates. Workers fan their wings to circulate air through the hive, actively lowering moisture levels. Over time, the water content drops from roughly 70% in nectar to below 18% in mature honey. This low water concentration is critical. Microorganisms require water to grow and reproduce. When water activity falls below a certain threshold, microbial metabolism becomes impossible. Honey's high sugar concentration creates an osmotic environment that draws water out of bacterial cells, effectively dehydrating them. Osmosis refers to the movement of water across a membrane toward higher solute concentration. In honey, the solute concentration is extreme. Bacteria cannot thrive. Honey's antibacterial properties extend beyond low moisture. During the enzymatic breakdown of sugars, another reaction occurs involving glucose oxidase, an enzyme added by bees. When honey is diluted slightly, glucose oxidase converts glucose into gluconic acid and hydrogen peroxide. Hydrogen peroxide is a reactive molecule capable of damaging bacterial cell structures. In diluted honey, small amounts of hydrogen peroxide are produced continuously, contributing to antimicrobial activity without harming the bee or, in moderate use, human tissue. Acidity also plays a role. Honey typically has a pH between 3.2 and 4.5, making it mildly acidic. Many pathogens prefer neutral environments. Combined acidity, low water activity, and hydrogen peroxide generation create multiple barriers to microbial survival. Stability is not the result of a single property, but of layered chemical defenses. Because of this composition, honey resists spoilage to an extraordinary degree. Archaeological discoveries have uncovered sealed jars of honey in ancient tombs that remain chemically recognizable after thousands of years. In the dry burial chambers of ancient Egypt, honey was placed among grave goods, both as food and as offering. Chemical analyses of such samples reveal preserved sugar structures and low moisture content, confirming honey's long-term stability when protected from external humidity. Ancient Egyptian texts and reliefs depict honey collection and use. Honey served as a sweetener, a preservative, and a medicinal substance. It was applied to wounds, mixed into salves, and incorporated into ritual practices. Its symbolic association with purity and abundance reflects not only taste but durability. A substance that does not rot easily becomes culturally significant. Archaeological residues of honey have also been identified in pottery fragments from various ancient civilizations. Through chemical fingerprinting techniques, researchers detect characteristic sugar ratios and organic acids that distinguish honey from other plant products. These findings suggest that humans have harvested and valued honey for at least several thousand years. The relationship predates formal agriculture in many regions. The preservation of honey over millennia is not magical. It is chemical. If exposed to high humidity, honey can absorb water from the air, raising its water content and enabling fermentation by yeasts. Properly sealed and stored in dry conditions, however, its composition remains remarkably stable. Time alone does not degrade it. Environmental change does. Within the hive, once the desired moisture level is reached, bees seal honey-filled cells with a thin wax cap. This capping isolates the honey from ambient air, maintaining its low water content. The wax barrier functions as both storage lid and humidity shield. Inside that sealed cell, sugar concentration, acidity, and enzymatic balance remain steady. Honey's texture can change over time through crystallization. Crystallization occurs when glucose molecules separate from solution and form solid crystals. This process does not indicate spoilage. It reflects natural sugar ratios. Gently warming crystallized honey dissolves the crystals without altering its fundamental composition. In chemical terms, honey is a concentrated energy reserve composed primarily of glucose and fructose, with trace enzymes, organic acids, amino acids, and aromatic compounds derived from flowers. In historical terms, it is one of the oldest preserved foods associated with human culture. The same biochemical processes that protect it inside a hive allowed it to survive inside tombs and vessels, long after the bees that made it had disappeared. What begins as a transient floral secretion becomes a stable archive of plant chemistry and bee labor. The transformation from nectar to honey is gradual, enzymatic, evaporative, and precise. It is a collaboration between insect metabolism and environmental control. Inside a sealed comb cell, honey rests in near stillness. Sugar-dense, water-scarce, acidity-balanced, hydrogen peroxide latent. Its resistance to decay is not dramatic. It is methodical. Through chemistry layered upon chemistry, time is slowed. Long before modern hives and protective suits, humans searched for honey in wild landscapes. Early beekeeping did not begin as management but as gathering. Rock paintings in Spain, dating back thousands of years, depict figures climbing to access honeycombs suspended from cliffs. These images suggest that honey hunting predates organized agriculture. At first, bees were not kept. They were located. Over time, methods shifted from extraction to stewardship. In ancient Egypt, cylindrical clay hives were stacked horizontally along the Nile. Beekeepers transported these hives seasonally, following the bloom cycles of flowering plants. Relief carvings show smoke being used to calm bees and honey being filtered through cloth. Even then, there was awareness that gentle handling preserved both bees and yield. In parts of ancient Greece and Rome, woven baskets coated in clay, known as skeps, were used to house colonies. These structures provided shelter, but made honey harvesting destructive. Often, the comb was removed entirely, and bees were displaced or killed in the process. Early beekeeping balanced sustenance with loss. Knowledge advanced slowly. Beeswax became as valuable as honey itself. During the Middle Ages, beeswax candles illuminated churches, monasteries, and royal halls. Unlike tallow candles made from animal fat, beeswax burned cleanly and with little smoke. Its flame was steadier, its scent faintly sweet. In religious contexts, beeswax carried symbolic associations with purity and light. Entire economies supported wax collection and trade. Another transformation of honey emerged in the form of mead. Mead is an alcoholic beverage produced by fermenting honey with water and yeast. Long before refined sugar was common, honey served as a primary fermentable sweetener. Mead production appeared in multiple cultures, from northern Europe to parts of Africa and Asia. Fermentation converts sugars into ethanol through microbial metabolism. Where grapes or grains were scarce, honey provided an alternative path to preservation and celebration. The turning point in beekeeping design came in the 19th century with the development of the movable frame hive. Lorenzo Langstroth, an American clergyman and beekeeper, identified what he called bee space, a precise gap measurement that bees neither fill with comb nor seal with propolis. By incorporating this spacing into wooden frames, he created a hive in which comb could be removed without destruction. The Langstroth hive allowed inspection, disease management, and honey extraction without dismantling the colony. Design shifted from fixed structure to modular system. This innovation reshaped apiculture, meaning the practice of beekeeping. Movable frames enabled selective breeding, controlled harvesting, and improved overwintering strategies. Colonies could be expanded, divided, or relocated more efficiently. Observation replaced guesswork. The relationship between beekeeper and hive became less invasive and more continuous. Hive design continued to evolve. Variations such as top bar hives and vertical flow systems emerged, each attempting to balance bee health, honey access, and ease of management. Materials shifted from clay and straw to milled wood, and later to insulated composites. Ventilation, spacing, and frame depth were adjusted according to climate and floral availability. Architecture adapted to environment. In recent decades, urban beekeeping has grown in visibility. Rooftop hives now appear in cities across Europe, North America, and parts of Asia. Contrary to earlier assumptions, certain urban environments provide diverse forage through parks, gardens, and ornamental plantings. Rooftop colonies often coexist with dense human populations. The presence of bees in cities reflects a renewed awareness of pollination and local food systems. City beekeeping also redefines scale. A single rooftop hive does not produce industrial quantities of honey, but it fosters local engagement. Schools, community gardens, and small businesses incorporate hives into educational and sustainability projects. Honey harvested in urban neighborhoods carries a distinct floral fingerprint shaped by city plant diversity. Honey trade, however, remains global. International markets move thousands of tons of honey across borders each year. Major producers include countries such as China, Turkey, Argentina, and Ukraine. Honey is graded, filtered, blended, and packaged for worldwide distribution. Trade regulations attempt to address authenticity and contamination concerns. Adulteration, the addition of sugar syrups, has become a regulatory focus in some regions. Global production statistics fluctuate with climate, disease outbreaks, and agricultural demand. According to recent agricultural data, worldwide honey production measures in the millions of metric tons annually. Managed honeybee colony numbers have increased in some regions due to commercial pollination demand, even as concerns about wild bee diversity persist. Production growth does not necessarily reflect ecological stability. It reflects management intensity. Cultural continuity in beekeeping is visible in language and ritual. Phrases such as "telling the bees," a European folk tradition in which families informed hives of births or deaths, reflect symbolic integration of bees into domestic life. In many societies, bees were considered household members. Their productivity and survival were linked to human fortune. Across centuries, the materials changed, but the exchange remained constant. Honey moved from cone to vessel. Wax moved from hive to candle. Fermented honey moved into cups and ceremonies. Wood frames replaced clay cylinders. Rooftops replaced riverbanks. Yet the central relationship endured. Humans provided shelter and protection. Bees provided sweetness and light. Beekeeping today spans hobbyists with a single backyard hive and large-scale commercial operations, transporting thousands of colonies for pollination contracts. Technology now includes hive sensors that monitor temperature and humidity in real time. Yet the essential structure remains recognizable: a box, a colony, a season of bloom. From ancient cliff faces to modular wooden frames, from monastery candles to rooftop jars, labeled by neighborhood, honey and wax have traveled through history without losing their identity. They remain products of patient labor, shaped by insect biology and human adaptation alike. Survival in bees is not only a matter of daily labor. It is written into genes, shaped by climate, refined by disease, and influenced by human management. Beneath the visible structure of the hive lies a quieter layer of continuity, inheritance. Honeybee populations are not genetically identical across regions. Colonies in Mediterranean climates differ from those in northern Europe. African subspecies exhibit behavioral and physiological traits distinct from temperate populations. These variations reflect local adaptation, meaning genetic changes shaped over generations by environmental pressure. Temperature, humidity, floral cycles, and pathogen exposure all influence which traits persist. Heat tolerance provides one clear example. In warmer regions, certain honeybee populations demonstrate improved thermoregulation and higher survival rates under elevated temperatures. These bees may forage earlier in the morning or later in the evening, reducing exposure to peak heat. Genetic differences influence metabolic thresholds and behavioral responses. Adaptation emerges gradually through selection, not design. Disease resistance also varies across lineages. Some honeybee strains show enhanced hygienic behavior, meaning they more effectively detect and remove infected brood. Others display improved grooming behavior, removing parasitic mites from their bodies. These traits are partly heritable. When present in higher frequency within a colony, disease spread can slow. Genetic variation acts as biological insurance. Modern apiculture often involves artificial queen rearing. In this process, beekeepers select larvae from desired colonies and rear queens under controlled conditions. Artificial queen production allows rapid expansion of colonies and propagation of favorable traits. However, it also carries risk. If a limited number of genetic lines dominate commercial breeding, diversity narrows. A genetic bottleneck occurs when a population's gene pool becomes restricted. This can reduce variability in immune response, stress tolerance, and adaptability. In honeybees, widespread distribution of closely related queens may increase vulnerability to emerging pathogens or environmental shifts. Uniformity simplifies management, but may reduce resilience. Selective breeding programs attempt to balance productivity with health. Traits such as honey yield, calm temperament, and disease resistance are considered. Yet selection is complex. Enhancing one characteristic may inadvertently reduce another. Genetic management requires careful monitoring to avoid unintended consequences. Beyond managed honeybees, wild bee species contribute critical genetic diversity at the ecosystem level. These include bumblebees, mason bees, leafcutter bees, and numerous solitary species. Each occupies distinct ecological niches and exhibits unique physiological traits. Some solitary bees demonstrate high efficiency in specific crops. Others show tolerance to local temperature extremes or distinct pathogen pressures. In regions experiencing rising temperatures, certain native bee species appear better adapted than imported honeybee strains. Their life cycles align closely with local flowering patterns. Their nesting strategies reflect soil type and seasonal rhythm. Adaptation at the species level reinforces ecosystem continuity. Wild bee populations also act as ecological buffers. If disease reduces honeybee colonies in a region, native pollinators may partially compensate for pollination services. This redundancy stabilizes plant reproduction. Diversity spreads risk across multiple biological strategies. However, wild bee populations face their own genetic challenges. Habitat fragmentation can isolate populations, limiting gene flow between groups. Reduced gene flow increases inbreeding risk. Smaller populations are more vulnerable to stochastic events, meaning random environmental fluctuations that can sharply reduce numbers. Conservation strategies increasingly emphasize native species preservation alongside honeybee management. Protecting nesting sites, maintaining floral corridors, and reducing chemical exposure support not only abundance, but genetic exchange. Genetic diversity requires both population size and connectivity. At the molecular level, survival reflects gene expression patterns responsive to stress. Heat shock proteins, antimicrobial peptides, and detoxification enzymes are regulated according to environmental demand. These physiological responses are influenced by inherited genetic frameworks. Variation in these frameworks determines the limits of tolerance. Genetics does not operate independently of environment. It interacts continuously with external conditions. A lineage well adapted to one climate may struggle in another. Managed relocation of colonies across regions can introduce mismatches between genotype and landscape. Artificial queen rearing, when carefully diversified, can preserve and distribute beneficial traits. When concentrated narrowly, it may accelerate bottleneck risk. Responsible breeding practices increasingly incorporate genetic testing and controlled mating areas to maintain variation. Continuity depends on both inheritance and diversity. A single strong lineage cannot replace a network of varied populations. Resilience emerges not from uniform strength, but from distributed capacity. Within each hive, eggs laid today carry fragments of past adaptation. Across landscapes, multiple species express different solutions to similar environmental pressures. Survival is layered: individual, colony, species, ecosystem. Fragility and continuity coexist. Genetic variation expands possibility. Narrowing that variation contracts it. In the quiet exchange between inheritance and environment, bees continue to adjust, generation after generation, balancing stability with change. The future of bees is not written in certainty. It unfolds through research, experimentation, and quiet adjustment. While environmental pressures remain complex, scientific inquiry continues to expand the tools available for understanding and support. Among the more visible innovations are robotic pollination projects. Engineers and biologists have explored small autonomous devices capable of transferring pollen between flowers. These robotic systems are not designed to replace bees in natural ecosystems. Instead, they function as research platforms, testing the mechanics of pollination and offering contingency solutions in highly controlled agricultural environments. The existence of such projects reflects both technological ambition and awareness of pollinator vulnerability. Artificial intelligence has entered beekeeping in more subtle ways. AI-assisted colony monitoring systems analyze patterns in hive sound, temperature, and activity. By detecting deviations from normal acoustic signatures, algorithms can flag potential issues such as queen loss, swarming preparation, or stress events. These systems do not manage colonies directly. They provide early signals, allowing beekeepers to respond with informed timing. Sensor-equipped hives extend this capability. Modern hives may include internal temperature probes, humidity sensors, weight scales, and even carbon dioxide monitors. Weight changes can indicate nectar flow or food depletion. Temperature fluctuations may reveal brood instability. Continuous data collection transforms the hive into a measurable environment rather than an opaque box. Data analysis enables earlier disease detection. Patterns in hive weight, sound frequency, and brood temperature can reveal anomalies before visible symptoms appear. Machine learning models, trained on large datasets, can identify correlations between subtle changes and later colony decline. Early intervention becomes possible when warning signs are recognized in advance rather than after damage is widespread. These technological systems do not eliminate biological complexity. They illuminate it. Monitoring does not replace management. It enhances perception. The goal is not to mechanize the hive, but to understand its signals more clearly. Beyond technology, ecological restoration remains central. Biodiversity gardens, planted with native flowering species, provide forage continuity across seasons. Such gardens support not only honeybees, but also wild pollinators with specific dietary needs. Diversity in plant species promotes diversity in bee species. Urban parks, roadside margins, and community spaces increasingly incorporate pollinator-friendly designs. Urban flowering initiatives have expanded in many cities. Municipal planting programs now prioritize nectar-rich species over purely ornamental varieties. Green roofs and balcony gardens contribute additional microhabitats. In some cases, urban environments with varied plantings and reduced pesticide use have supported stable pollinator populations. Cities, once considered ecological voids, can become structured refuges. Community-supported beekeeping has also grown. Local cooperatives and educational programs connect residents to hive stewardship. Participants learn about seasonal cycles, disease management, and pollinator ecology. This distributed knowledge strengthens public awareness and responsibility. Bees move from abstraction to presence. Scientific monitoring networks operate across regions and continents. Researchers track colony losses, pathogen prevalence, pesticide residues, and climate patterns. Long-term datasets reveal trends not visible in single seasons. Collaboration between universities, government agencies, and citizen scientists expands observational reach. The scale of monitoring mirrors the scale of the challenge. Importantly, future resilience does not depend solely on honeybees. Conservation strategies increasingly recognize the importance of protecting native bee species. Planting diverse habitats, preserving nesting sites, and reducing chemical exposure support a broader pollinator community. Stability arises from multiplicity rather than singular reliance. Technological tools, ecological restoration, and community engagement intersect. Sensors generate data. Data informs management. Gardens provide forage. Monitoring networks track outcomes. The approach is layered rather than linear. Hope, in this context, is not dramatic. It is procedural. It exists in incremental improvements, in refined breeding strategies, in improved habitat corridors, in informed pesticide regulation, in restored flowering margins along agricultural fields. Each adjustment reduces pressure slightly, expanding the margin of resilience. Bees have persisted for millions of years through climatic shifts and ecological change. Their current challenges are linked closely to human systems. It follows that human systems can also become part of the solution. Quietly, through design choices, planting decisions, monitoring technologies, and collaborative research, the relationship between bees and the living world continues to evolve. Across this journey, from the architecture of a single body to the chemistry of honey, from the logic of the colony to the pressures of the wider world, one pattern remains consistent. Continuity depends on balance. Balance between diversity and adaptation, between management and wildness, between observation and restraint. As night settles, hives rest in layered warmth. Outer bees form insulation, while those inside maintain steady heat through quiet muscular vibration. Sensors may record slight shifts. Data may travel across networks. Yet within wooden walls, regulation continues without spectacle. Honey sealed in wax holds stored sunlight. Bodies rotate gently. Balance is sustained through repetition, not force. The future of bees is not secured by a single breakthrough, but by steady attention through careful breeding, restored habitats, restrained chemical use, thoughtful monitoring, and shared responsibility. Progress moves in small adjustments rather than dramatic turns. Fragility remains part of the story, but so does continuity. In the quiet persistence of each generation, adaptation continues.